Good morning, everybody, and welcome to our first trip here at the San Diego chapter for this fiscal year. I want to welcome you all to a bright and sunny and warm day in San Diego. Wish you could all be with to be able to do this virtually. And as I said a little bit ago, we are so proud and pleased to be able to focus on our San Diego Zoo here this morning and showcase not only the zoo, but our three speakers are all ARCS alumni. And that's very special as well. They all happen to be from UC Davis. So a big kudos to the Northern California chapter for making them scholars. And then we also have one of our current scholars who's also going to join the presentation this morning. And so I just want to invite everybody to enjoy ARCS in action at San Diego Zoo Global. And today we're going to be talking with our four scientists about removing diseases as a roadblock to wildlife conservation. And we're going to be hearing from the three scholars, former scholars now, scientists at the zoo that are part of the disease investigation group. And of course, what's really made this even more interesting and timely right now is that we're dealing with a global epidemic that started in animals and moved to people, COVID-19. And so there will be some discussion about that. So at this point, I would like to turn the program over to Ann Siemens. Ann is one of our long-term members who incidentally was an executive at the San Diego Zoo until she recently retired. Ann is going to introduce the speakers, the program, and thank you for joining us. Welcome. And Ann, let's roll it out.
It's such a pleasure to be with everybody, and especially a pleasure to introduce our wonderful ARCS scholars and alumni. When I first came to the San Diego Zoo, I was impressed by, number one, the fact that there was a Beckman Center there. I had worked with Dr. Arnold Beckman for 20 years and knew that, in fact, his daughter, and he told me when the Beckman Center was being funded, it became a favorite place of mine to go. Getting to know the scientists was a real gem of an opportunity. And to learn that there were three ARCS alumni in the disease investigation unit says a whole lot for the science going on at the San Diego Zoo. Few people understand, or maybe have even heard, that the San Diego Zoo is the only zoo in the world that has a multidisciplinary research program that is zoo-based. And that's one of the distinguishing differences that will help the zoo moving forward, as we so badly need to conserve species, understand diseases, and keep our world intact. So I'm pleased to introduce to you our three speakers and our current scholar who will be presenting this, Dr. Patricia Gaffney. I'm not going to read all of their credentials because that would take up the whole time, but our speakers are both DVMs and PhDs, among other things. Dr. Gaffney will be our moderator, and she is the senior scientist in disease investigation and works as an anatomic pathologist and researcher, providing diagnostic support for the San Diego Zoo Global's animal collection and field conservation programs. She manages collaborative pathology investigations for both field programs, and her research investigates the genetic basis of disease in endangered species. She earned her bachelor's degrees in animal pathology and neuroscience from University of California, San Diego, cum laude, I would add, Phi Beta Kappa. She received her doctorate in veterinary medicine and master's in preventative veterinary medicine from UC Davis. And she was a Steele Endowed Pathology resident and board certified and is board certified in anatomic pathology. Oh, golly. If I went on with all of her honors, it would take a long time. So I'll leave it there and move on to Dr. Stephen Kubiski, who is also a senior scientist in the Disease Investigation Unit at San Diego Zoo Global. Steve serves the San Diego Zoo Global in disease investigation, and in his role, he works with anatomic pathologists. He works as an anatomic pathologist, providing diagnostic support for wildlife disease investigations in the animal collection and field conservation programs. He works in the molecular diagnostics laboratory to facilitate investigations of infectious disease that affect the Institute's conservation programs. He works with implementing new diagnostic and research tools in the molecular diagnostic laboratory. And he earned his bachelor's degree in biology and his doctorate in veterinary medicine from the University of Georgia, followed by a year as a wildlife disease diagnostician in the Southeastern Cooperative Wildlife Disease Study at the University of Georgia. He then entered a residency in anatomic pathology through a joint program between UC Davis and the San Diego Zoo, where he was the Steele Endowed Pathology resident. Following board certification, Steve entered a PhD program in integrative pathology at UC Davis, where he recently concluded his dissertation researching a newly discovered canine virus. He's a diplomat of the American College of Veterinary Pathologists, an alumnus of ARCS, and an active member of the Wildlife Disease Investigation. So we look forward to hearing from Steve. And his area of molecular diagnostics has a lot of exciting opportunities that are coming onto the field of conservation and disease management. Also, we have Carolyn Moore, also PhD, DVM, and she currently is the Steele Endowed Pathology Fellow. She serves the San Diego Zoo in this role, working as a veterinary toxicology researcher, providing molecular and diagnostic technology support. She uses toxicology, pathology, molecular diagnostics, and epidemiology to better understand how environmental contaminants, such as heavy metals, pesticides, and harmful algae blooms, create roadblocks to wildlife conservation and how to prevent them. Carolyn earned her bachelor's degree in biochemistry and molecular biology from the University of California, Santa Cruz, assisting with necropsies and research on the decline of the southern sea otter. She earned her doctorate in pharmacology and toxicology and her veterinary medical degree at UC Davis, while on an Environmental Protection Agency STAR grant. So we've got a very distinguished presenter group, as well as a current ARCS scholar, Tyler Mather, who is currently a graduate student in biology at the Shikuma lab at San Diego State University. This lab studies the human microbiome. There's a lot of crossover here between the human microbiome and the microbiome work that is being done at the San Diego Zoo, and a lot of the toxicology work and diagnostic work that you hear from the zoo researchers are the kinds of things that are used in microbiome work that, such as Kyle is doing. So welcome to all three. We're very eager to hear you talk, so I will quit talking. Thank you. Patty, take it away.
Thank you very much, Anne. Thank you, Holly, and thank you to ARCS for inviting us to give this virtual tour of our department, Disease Investigations, here at San Diego Zoo. We wish we could meet you all in person, and hopefully we will someday, but for now we've prepared a presentation to kind of walk you through who we are and the work that we do. So just to reiterate, we'll give some brief introductions to ourselves, complementing Anne's introduction, and then talk to you about some examples of the specific kind of work that we do. So again, I'm Patty Gaffney, and I started off my work in conservation right here in San Diego. As Anne said, I have a bachelor's degree from UC San Diego, and it was during that time that I actually started working at the San Diego Zoo, first as a volunteer and then as a scientist hired to do behavioral observations on white rhinos. And this is one of my prized pictures of the 2 last female northern white rhinos at the safari park, who are sadly no longer with us. This is Nola and Nadi. And this was my first introduction to conservation work and really one of the driving forces in my career. So I worked doing these behavioral observations as an undergrad and then moved on to veterinary school at UC Davis, where I worked hard to get my hands on as many animal species as possible. I did get my Doctor of Veterinary Medicine from UC Davis, as well as a master's degree in preventive veterinary medicine, which is an epidemiology and population health-based degree. And it was during that time that I really developed my interest in understanding the mechanisms of disease and how you apply that on a population level, which is what drove me into a career as an anatomic pathologist, which again was a crossover program between UC Davis and San Diego Zoo, and culminated in a board certification in anatomic pathology. Following that, I actually returned to UC Davis, first as a instructor for veterinary students and residents, but then ultimately pursuing my PhD. And that is where ARCS came into my life, and gladly I was happy to have funding from ARCS for 2 years of my PhD dissertation work, and that was through the Northern California chapter. And this is just a little example of the work that they funded. So I worked on an endangered species, the island fox, which is native to the Channel Islands off the coast of Southern California. And through this work, I studied a rare disease normally, but it ended up being a common disease of foxes. And I was interested in understanding the risk factors for disease in that population. And so that work culminated in several publications, and ARCS supported my travel to various meetings in order to present this work at international conferences. Following my PhD work, I actually, again, went back to UC Davis. And actually, I should say that the majority of my PhD work took place at UC San Diego through the School of Medicine with a joint faculty advisor there. But after that was completed, I returned to the UC Davis veterinary school world and worked as a pathologist for the state diagnostic lab, looking at infectious diseases, herd health, and wildlife diseases. But then that brings me full circle back to San Diego, where about 3 years ago, I was recruited into a position of senior scientist. And in my role here, I do spend a lot of time at the microscope. You can actually see I'm sitting next to my microscope right now. But as our pathologists for field conservation programs, they also let me out of the office. And I do get to spend quite a bit of time in the field. I have a postdoctoral fellow in Kenya, who you can see pictured here in the top middle of the screen. And I get myself into some interesting situations trying to be a pathologist working out in the field. So now I'll turn it over to Caroline to introduce herself.
Yeah, happy to. And just to let you know, Patty, I think you might have touched the screen again. If you want to open and close to get rid of those. But I'm Caroline. As you can see right now, I haven't left California. I'm pretty attached to the state. I like to think that my love for conservation wildlife actually started as a baby, because I spent the first 2 years of my life on a wildlife refuge in Elkhorn Slough. Before moving up to Eureka, where I fell in love with actually chemistry and nature, and I knew I wanted to be a veterinarian. So I went to UC Santa Cruz with that in mind, but ended up falling again in love with chemistry and did biochemistry and molecular biology as an undergrad. And as I went through that process, I was working in a small dog and cat clinic, and I was working for the Department of Fish and Wildlife, looking at the decline of the southern sea otter, and looking at a few different pathways of how that might happen, as far as the spread of pathogens and antibiotic resistance, and a toxin called microcystins. That's actually a natural toxin produced by algae. And so I was thinking about all these parts together, and found out that actually UC Davis has this dual degree program to do a PhD in DVM, and also has this huge One Health component to it. Fell in love with it and ended up doing my PhD in pharmacology and toxicology at UC Davis. And that's featured here in this picture with an actual C. elegans worm here because I use an alternative model to see if these toxins could be neurotoxic or not. And then as I went through the PhD side of things, I didn't focus too much on wildlife. I did more, some with livestock, some with equine. Still was interested in this cross-species comparison part. And when I was finishing up my DVM, I decided, like, do I want to try to get back into research, and ended up going to UCLA for a postdoc. And that's when I fell in love with this idea that, well, I don't know if love's the right word, but was really intrigued with this idea that you could be exposed to something like when you're in utero and developing from your parent and never see that environmental contaminant again, and that could have lasting effects over your life. And so playing with this idea and thinking about wildlife conservation, I was realizing this is a feature that isn't communicated that often as we look at our species populations, and reached out to Steve Kubiski, who will be next, who is actually now my supervisor, to see if they'd be interested in toxicology and wildlife conservation. And there was a lot of links there as far as what the animals were experiencing and, you know, the need for research moving forward. So I was very excited to start my position here at San Diego Zoo Global about a year ago. So here's just some fun pictures. The one where it says UC Santa Cruz, that's actually me at like 3 in the morning digging innkeeper worms. And so this was kind of when I realized I wasn't going to be a straight biologist, because I wasn't that into getting super muddy at 3 in the morning. But it was fascinating to be out there and finding this link where, you know, as we look at what our sea otters being exposed to, looking at their food sources and seeing if those food sources are getting contaminated, it really helped bring the picture together. There's a picture of me at UCLA lab where I was training up benchtop research again. And then coming at the bottom is where I used to work with a mentor to do low-cost clinics. So we just neutered that little mini pony. And then I haven't done a lot of field work because COVID, but I was able to go release some kangaroo rats recently. So that was super exciting. Okay, next slide. And throughout this process, ARCS really stepped in not only supported me as a fellow, but I had the opportunity to present to the Northern California chapter, I think in 2012. So I presented this talk, Algal Toxins and Emerging Health Concerns, because these algal blooms occur often when there's slow moving water and climate change and increased nutrients. So it's something that's building all over the globe that we'll talk about more later. And I was looking at ways to detect these toxins, and that led to this first paper that ARCS helped support, and the methodologies that we developed there, we actually applied to the second paper listed here that was looking at a pug that was eating a supplement and having liver disease. That turned out it was because of this human grade supplement it was eating. And then that was also scary because the third paper listed here shows that some of these toxins that we think of being you know, you need a pretty high dose to get an effect, actually had neuro effects at low doses. So it was amazing that ARCS not only was interested in my research, but wanted to hear about it and was able to support a lot of this work moving forward that is still supported within the veterinary conservation world today that I've been working on. Thank you. Next slide.
All right, and now Steve will take it away.
All right, thank you. I am the token redneck here, I guess. I am not from California. I grew up in Georgia, where I did my undergraduate and veterinary school at the University of Georgia, where I specialize sort of as much as you can on population health and pathology. So most of my senior rotations were either diagnostic pathology or wildlife related externships. I had to dig up a few of these pictures to remember that I actually did at some point know how to deal with a stethoscope and live animals, which is kind of fun. I miss it. After graduating vet school, I worked as a diagnostician at the Southeastern Cooperative Wildlife Disease Study, which is based at UGA. It's a large research and diagnostic facility exclusively dedicated to wildlife diseases. So they contract with, I think at this point, over 20 different state wildlife agencies. So whenever those biologists want to investigate a single death or a mortality event in a wildlife species, they submit them to the SQUIDUS, we call it, for an investigation. So I got to do a year there. After that, I came out to California to do my anatomic pathology residency. Patty, I think you can, oh no, you can leave it there. Go back. I'm not ready. For my residency, which was a joint program, just like Dr. Gaffney did between the veterinary school at UC Davis and the folks down at the San Diego Zoo, where we spent our third year. Okay, next slide. After I took my board exam, I went back up to Davis to the graduate group in integrative pathobiology for my PhD. I was looking for pictures, and I found this video actually on, I can't even remember what forum, but it was a video from the first Meet the Minds of the Future symposium up at Sanford that ARCS put on. And so it shows the area of my dissertation research, which was investigating and characterizing a newly discovered virus that we found in dogs. And I was also an ARCS scholar for 2 years, so they really are instrumental in contributing to this. And when I would give talks or attend to these ARCS functions, it's really important to note how the flexibility of ARCS funding is so helpful, because oftentimes you'll get money from other sources and it's so strict in the way it can be used. So the ARCS money being able to fund both research efforts and materials, and also contribute to stipend for these starving grad students is really amazing. So the work that I did during my PhD is actually really similar to the type of independent research that I do down here at the zoo, which is viral discovery and trying to characterize diseases that may be associated with any newly discovered viruses. Next slide. And I just did want to mention that one of the chapters of my dissertation up at Davis was on the discovery and characterization of a novel virus of red pandas. At the time, there was a current pathology resident working on this in our lab. And after, just like me, he went back to Davis after his residency, and now he's doing a PhD on this virus in my old lab. And actually, he just recently received his first ARCS scholarship. So now he is an ARCS scholar in the Northern California chapter working on this virus that we started years ago. And I'm actually kind of co-mentoring him in this project. So we're really excited about that. Next slide. So after 35 years of school, I finally got a real job. I was lucky enough to come back down to San Diego and get a permanent position with the disease investigations group here at the zoo, where I get to do what I really want to do, which is go on sundowner safaris in Africa and take these overdramatic photos in the field. And you can go to the next slide. Since Patty mentioned a virtual tour, oh, you changed my picture. That had a story behind it.
Oh, sorry. It was blurry.
So we all work in the same group, which is disease investigations, which is why we wanted to give a little background about this group to put our work into context. So the research at the San Diego Zoo, as Anne mentioned, goes back to the 70s with the formation of CRESS, which stands for the Center for Reproduction of Endangered Species. It was based at the Scripps building, which is downtown, a little building that's attached to the veterinary hospital right behind the Old Globe Theater. You can see Dr. Bernershka there in the center in the back, and there's actually a few people in this photo from 1975 that are still kicking, working with us today. Next slide. This is about 20 years later. You can see how much larger the group is. They grew and they diversified from not just reproductive work, but also genetics and infectious disease at the time, among other disciplines. And many of these people are also still working with us today. Next slide. So this was last year, probably early last year, just to indicate how much our group has grown. The folks in this picture probably represent maybe half of the people that work for the Beckman Center on a day-to-day basis. And we, of course, have moved from the Scripps building downtown to this really state-of-the-art facility called the Beckman Center, which is up at the Safari Park. And we now consist of 8 different major branches of research that you can see here in this list. And of course, Caroline and Patty and myself are part of the disease investigations group. Next slide. So our tagline, our mission statement, I should say, is removing disease as a roadblock to conservation. Go ahead. And we also have grown quite a bit from our sort of humble beginnings. I think this photo was about the same time back in the mid-90s, where it was just 3 folks in the pathology department. And then this current photo is fairly current. I think it's last year. But we've got about 20 people now, and that represents pathologists and technicians, people working for the pathologist, people working in the molecular diagnostic lab, and then all of our support staff. The next slide. So I just want to break down, Patty mentioned a virtual tour, which I think is a good term. I just want to break down what disease investigations consists of. we kind of have, I'm putting the word divisions in air quotes because we're not really that separate and distinct. We're all working together in overlapping objectives on a daily basis, but it kind of helps to break it down this way. So the first is the Amphibian Disease Lab, which was developed originally in 2006 with just a little bit of seed money, followed by a larger grant in 2008. And the whole point was to provide cheap and rapid diagnostic testing for chytridiomycosis, which is a fungal disease that has decimated amphibian populations all over the world. So there was a really, really urgent need to be able to test for this so there could be management and mitigation strategies put in place. So this is sort of our flagship research in our lab. And we do continue to receive samples from all over the country and even other countries to test for this fungus. So those organizations dealing with amphibian conservation can have some of that cheap, quick support. Next slide. So it does go to show what a little seed money can do. Our lab has grown substantially since then to include not just the testing for that chytrid fungus, but also samples that we have through contracts with the U.S. Fish and Wildlife Service to test for numerous pathogens from other species and projects on endangered species, especially in Southern California. Additionally, as pathologists, no, go back. Additionally, we send up samples to the lab as part of our own individual diagnostic investigations. If we're suspicious about a certain infectious disease, we can just send samples up to the Beckman Center. And we provide service to the clinical veterinarians from the zoo and safari park as well with the same service. So all of those vets working on the little critters on a day-to-day basis, they routinely use our lab as part of an annual health exam, testing as certain illnesses, and then part of both shipping and receiving animals from other facilities as part of just routine biosecurity. And we're also always developing and improving the assays that we offer. And we've always had various independent research projects going on in the lab as well. So I mentioned that I like to do viral discovery and disease characterization. And one of the more high profile projects that's going on in that lab, Dr. Gaffney is going to get into in a lot more detail in just a minute. All right, next slide. We also have our very own epidemiologist. So she studies patterns in data to make conclusions about things like how diseases are transmitted, who may be most at risk for certain problems, and what other factors such as time of year, location, weather, any of these things that might contribute to disease dynamics. And when you're studying diseases in a natural setting as opposed to an experimental one, epidemiology is critical to making conclusions that determine and affect how we can manage, treat, and prevent issues in the future. So we're really lucky to have Dr. Carmel Whitty, who's our epidemiologist, helping us with both captive management as well as our field projects. Next slide. And then last but not least, anatomic pathology. This is essentially our core function. As anatomic pathologists, we're basically diagnosing diseases by doing examinations of tissues, both macroscopically and microscopically. There's features that we can determine whether it's an infectious disease, a metabolic or nutritional disease, and of course things like cancer, trauma, allergic reactions, really anything. We can tease it out with our handy scopes. And just as a plug for zoo and exotic pathology, on any given day, we might receive anything from a giant stick insect to a gazelle, a lion, a fish, a frog, a snake. It's very, very interesting work. And we're one of the very few places in the world that gets to investigate exotic animal diseases to this level. Next slide. We've got archived microscopic slides that date back to the 30s, which equals, I think it's probably up to about 650,000 microscope slides. This slide archive is actually an accredited museum. And we tallied it up when we were hoping to digitize this archive by scanning these microscope slides into high resolution files that could be used for research and teaching. If we did that, it would equal 2.5 petabytes, which is a word I'd not heard of before, but it's equal to 2,500 terabytes of data. So this is an idea that we're constantly hoping to achieve, the quote unquote digital zoo. It would be a really massive step forward in using this gold mine of an archive for our own research, but also training other conservation scientists that hope to use anatomic pathology and disease investigations in their research as well. Next slide. So that's basically how our day-to-day breaks out within our group. But of course, the real story here is conservation. We're working for the health and well-being of our animals at the zoo and the park as ambassador species for their wild counterparts. That sort of defines what we call ex situ conservation, which is protecting a species outside of its natural habitat. In addition to the work that we do, of course, there are other, go back. There's other groups at the zoo that are helping, educate people about threats to endangered species, learning about animal health and welfare and behavior in a way that can always be extrapolated to defending them and protecting them in the wild, which, okay, now next slide. That is in situ conservation. So in addition to the captive animals, we are always providing direct support to wild populations by, through pathology, support to a variety of field programs. I did mention the testing that we do in our molecular lab for groups like the U.S. Fish and Wildlife Service, but our own San Diego Zoo colleagues have numerous field programs that we support through pathology, like the Pacific pocket mouse, terns and plovers, burrowing owls, and the endangered Hawaiian forest birds. Next slide. We also contribute a lot to conservation research through samples. We collect tons and tons of samples. We save lots of tissues at -80 degrees that can be used for anything from infectious disease research to vitamin and mineral analysis for both prospective and retrospective projects. We've got standing requests from probably thousands of species that when collected post-mortem, we send these tissues to the conservation genetics lab, and those serve as the sources for cells that end up making up the frozen zoo. And we also have a long list of requests for certain, especially the more rare species, to save for universities, museums, and other institutions. We can perform post-mortem exams in we call it a cosmetic fashion, so that the specimen can then be sent to the museum and be displayed. And so we like to say that sort of as an institution, the zoo does every animal there quite a bit of justice, not only by ensuring it's got the best environment and veterinary care, but the way we're able to further their contribution as ambassador species by immortalizing them through our microscopic archive, all the valuable research samples, and all the museum specimens. And I think, next slide, yeah, I will pass it back off to Patty to talk about some of this field work.
Yes, as Steve mentioned, we have this incredible archive, tremendous ability to do retrospective work and use those samples to also apply to new technology. But one of our strengths, just reiterating, is our multidisciplinary team that is poised to respond to disease threats in real time. And I'm just going to give you 3 brief examples of how that works. So as mentioned, the amphibian lab was developed to test for a severe fungal infection that has caused amphibian decline, chytridiomycosis. And this is something that we do routinely in an attempt to help save this species. So the mountain yellow-legged frog is a species, an endangered aquatic amphibian that is currently exists in fragmented populations throughout Southern California. And at the Beckman Center, we have a group of biologists, ecologists who do captive breeding of this frog and then release it back to the wild to what we hope are clean, pristine habitats that don't have a risk for chytrid. But unfortunately, sometimes we do get animals that do die in the wild, and those are collected and sent to us at the in necropsy, and we do postmortem exams, and we test all of them for chytrid. And we do actually see it in the wild quite frequently. We are able to give that information back to our field biologists basically in real time, so that they know if an area where they might want to reintroduce a species has chytrid infection. And this helps both plan for releases and helps us better understand the distribution of the disease in the wild. Another example of a species where we were part of a real-time response was a recent release of the Maui parrotbill, or the kiwi-kiu, as it's called in Hawaii. And this is a critically endangered Hawaiian forest bird, and less than 500 of these animals exist, and they only populate a 30-kilometer square region on the northeastern slope of a volcano on Maui. So as part of their species recovery plan, this animal, it was recommended that a second population site be made so they didn't just exist in one region. And so a lot of work went into designing the site with infrastructure, removal of non-native predators, and mosquito mitigation. And recently, I think it was towards the end of 2019, a group of kiwi-kiu were released in this site. But unfortunately, the animals, once released, started to become sick, and unfortunately, some of them did die. Where we come in is that the state of Hawaii can actually mail us a dead bird, and we can get it within 24 hours and do a postmortem exam. And in this particular case, we were able to diagnose the cause of death in this animal basically real time the same day, because we did what we call impression smears of lung tissue, and were able to identify a blood parasite that was in excessive numbers and was the cause of death of these birds. And we were able to give that information back to the biologists on the ground in real time, so that they could then implement management strategies to try to save the remaining birds. This particular parasite is called Plasmodium, and this is what is the cause of avian malaria. And because we were able to detect it in these birds in this location, it might change the site where additional translocations could take place. And then lastly, another example, this is an ongoing investigation, the San Bernardino kangaroo rat, another endangered species endemic to Southern California that is population is fragmented. These animals are meant to be translocated from a site that was actually contaminated with heavy metals because it was previously a gun range. And when these animals were caught up for translocation, it was noted that they had an abnormal hair loss patterns, and we didn't know why. And so a comprehensive effort, including veterinarians, pathologists, toxicologists, clinical laboratories, captured 7 of these animals from the wild and took multiple samples, including blood, hair, skin, feces, to try to understand what the cause of hair loss was. And this is an ongoing investigation, and we actually don't know what the cause is yet. But what I can say is that we were able to provide back information to the biologists on the ground to rule out that this was an infectious disease. So in preparation for translocation, we at least know that we're not moving an infection from one area to another, but we don't yet know the cause of what the cause of the hair loss. So we do provide pathology support and diagnostics in real time, but we are also part of larger research projects. One of the projects that is consuming a lot of our time and a lot of our molecular lab staff at the moment is trying to understand better the elephant endotheliotropic herpes virus that you may have seen in the news last year. This is a herpes viral infection that is known to be present in elephants, in African elephants and Asian elephants, but in African elephants, it's almost exclusively thought of as a benign infection that doesn't cause disease. However, last year in 2019, 3 African elephants in collections died, and 5 became severely ill and required aggressive therapy in order to recover them from this disease. And this was a big shock to the African elephant veterinarian research community, because in the previous 25 years, only 2 African elephants had died of this disease. So we wanted to understand why. And the disease is a mouthful, elephant endotheliotropic herpes virus, but all that really means is it's a herpes virus that attacks the cells that line blood vessels. And we'll just call it EEHV from now on. But because San Diego has such a large team, and we have a multi-generational African elephant herd at the safari park, we have undertaken a years-long, is actually multi-year research study aimed to understand better why or what are the causes of, or what are the factors that are associated with this virus being present in the blood and being shed in a variety of ways, either through the trunk or through saliva or through feces. So we have brought together our veterinary staff, our animal keepers, our epidemiologists, our molecular diagnostic lab, as well as collaborating with behaviorists and endocrinologists to try to understand what are the risk factors for this disease and how do we prevent it in our collection so that we don't have additional African elephant deaths. And this is something we're able to do because we have this large collection under managed care, but this disease also exists in the wild. And so once we develop our diagnostic recommendations for collection elephants, we also want to translate this into the field. So we do a lot of work, especially in Kenya, and in particular at an elephant orphanage called Reteti, where Again, we have Dr. Stephen Chege there in the bottom middle, our postdoc in Kenya treating an orphaned elephant calf. We know that this disease exists in the wild, but we don't understand how it manifests in wild elephants. And so we want to translate our diagnostic capabilities and our research study to the field and get a better understanding of the natural history of this disease. And just to highlight a little more of our field work in Kenya, this is a species you may have not heard of, a Horola antelope. And this is a species that's was new to me 2 or 3 years ago, but now is near and dear to my heart. This is the most endangered antelope in the world. And it exists only in as part of the native range of Horola in the southeastern portion of Kenya. And we work with a sanctuary, the Ishak Bini Horola Community Conservancy, to try to help preserve this species and also to understand its risks for disease. And so again, a photo from Kenya with our postdoc, Dr. Stephen Chege, who was recently able to participate in the collaring of 5 Horola, which we will now be tracking their movements in the wild to better understand their risk and exposure to disease. And we think that the disease risk largely comes from the surrounding livestock that share the same community and the same habitat with the Horola. And so to help try to protect Horola, we also have a vaccination campaign that occurs annually where we try to vaccinate livestock to create a buffer zone around the Horola sanctuary to prevent transmission or spillover from disease from livestock to the Horola. And just to wrap up, a lot of our international work is driven by the needs of our partners on the ground in these countries. And we're doing a lot of work trying to build capacity by establishing laboratories, providing equipment, providing training to our colleagues on the ground so that ultimately they will have laboratories established with the diagnostic capabilities similar to what we have, and they will be running those laboratories themselves. And I think then we will move on to Caroline to talk a little bit more about her work.
Yeah, so as you can see, disease investigations is incredibly wonderful at their infectious disease investigation part of it, but there's this idea of toxicology. And so obviously if there was an animal that had a suspected toxicosis, the pathologists are very equipped to work that up. But moving a step further into research and field application is where I come in. So I'm just gonna highlight a couple cases of where I've started to integrate myself at San Diego Zoo Global. So this is a picture of Kenyan vultures. On the left side, you see what typically happens when there's a carcass. So these are scavengers, they eat dead animals. And you can expect several different species of vultures, jackals, eagles, that sort of thing there. Something that does happen in Kenya and around the world is the accidental and on-purpose poisoning of these scavengers. So 2 scenarios where this might happen is if, let's say, a lion comes in and eats some livestock and kills it, and obviously that has a huge impact on the owners of that livestock. And so what they might do is lace that piece of livestock with enough pesticides to ideally remove the threat of that lion from killing more livestock. But what also often happens is that any scavenger that comes by throughout the day and night will also die. And so you can get these mass pileups of vultures, which is devastating because this is a species that has a long reproductive life and takes a while to rebuild these populations. The second example might actually be related to poaching. So as you know, vultures like to circle in the air and can highlight where a dead animal is. And so if the poacher doesn't want to highlight that, they'll actually lace the carcass of the elephant, which is not only devastating, but if you think about the size of an elephant, you can have hundreds of birds at these sites. And so you can lose 400, 500 birds at once. And so we're working to support Kenya wildlife toxicology, enhancing capacity, like Patty said, through workshops with people around the globe that study forensics and toxicology in wildlife. And this work not only impacts Kenyan vultures, but there's Andean condors and there's California condors and many species that are impacted by toxicology that we can take those tools and apply other places. Next slide. The next little story I wanted to throw out there was this idea of algal blooms, because this is something, like I said, near to dear to my heart that ARCS helped me support throughout my thesis, but also a concern that's growing around the globe. So as we get warmer, more stagnant, more polluted waters, we're more likely to get these blooms. And the blooms themselves aren't necessarily toxic. They have some side effects like using up oxygen and that sort of thing that could lead to, let's say, a mass fish die-off. But some of these blooms can actually produce tons of toxins. And so here on the left-hand side, we have a picture of Klamath River up in Oregon, and you can see the beautiful, almost paint-like picture that some of these blooms create. And then you can see that that similar color is kind of really striking in this image in the middle for the elephant scene. And this is actually, I don't know if you've been following the news, but there was about 350 elephants that died in South Africa that they're reporting is linked to cyanotoxins. And then this is just a highlight of like, this hits all species. This is actually a cow that has been drinking cyanotoxin containing water. And this particular toxin targets the liver. So you can see that his white patches of his skin is cracking because it can no longer handle the UV radiation, where the black patches are okay. So it really is across the board from California. I have California, South Africa to Australia, because Australia also has big blooms, but really globally. And we're working to figure out, you know, testing does exist for these types of toxins, but they're not set up so that maybe you're in a field setting where you have to have a ranger collect a sample and then hold on to that sample for 5 days and then ship it to the lab and they ship it somewhere else. So what can we do to preserve these samples and do better testing at source is like a field that we're really pushing right now. Next slide. And then the third case I was going to bring up here is the California condor, because we're all very proud of our California condors, and how, you know, we've really brought back the species to the wild. And there was a thought that if we, you know, not me, but if they work on reinstalling or reintroducing condors to marine habitats, that the carrion or dead animals that they eat would be less likely to be exposed to lead, which is the main concern with the inland dead animals, because though it's not zero, there is usually less lead shot in marine mammals. But those other concerns started popping up where marine mammals hang out obviously in the ocean. And I don't know if you guys are familiar with the fact that on the California southern coast, we have some of the highest levels of DDT ever in the world. And this is a toxin that likes to bind fat and hang out. Marine mammals have a lot of blubber and other sorts of fatty tissues, and so there's a concern that these legacy pesticides are accumulating in the marine mammals and then exposing the California condor there, which would have impacts on its reproductive health. So we're helping to look into this a little bit more, and I'm actually using some of the California condor type samples to help look at how scavengers in general handle some of these toxins, and then we can apply that back to the work in Kenya with the Kenyan vultures too. Next slide. And this is just, because I always like teaching a little something. Something that I think is really interesting right now and a hot topic is epigenetics and health. And so epi means on top, genetics is your DNA. So epigenetics is this field of manipulating your genome, your DNA in a way that doesn't actually change the genome. So here you have these little green stars that has some sort of modification that says, here, pick me, gene 1. And then there's other modifications that hide gene 2. And this has a lot to do with how the DNA is bundled and has access to be transcribed and translated to proteins to do some sort of cellular function. And so stemming back to work I did in my thesis and for my postdoc with this multi-generational thing, there's concerns that what the parent is exposed to, and here they're emperor tamarins that I drew, does it have a lasting effect each generation? And so are there things that we could pick up, like maybe skin color or tail color change, that is indicating that something's changing over time? And there's a data for this. So if you take, let's say, a mouse that's yellow coated and feed it a diet that changes its DNA methylation, which is a type of epigenetics, you can actually change the hair coat of the progeny. So hair coat might not matter that much, but what if this is changing its immune function? What if that's changing this ability to produce antibodies? Does that mean that maybe generation 2, 3, 4 can't fight off a virus as well? So these are some of the questions we're diving into moving forward. Go next slide. Especially in the lab at source. So Dr. Kubiski mentioned this idea that we're doing, and also Patty mentioned a lot of what we're doing in situ or at source. And so I'm helping with build the toxicology side of that, especially in disease surveillance. So this is using this idea that wildlife could be sentinels or, you know, help identify areas of concern. And not only can we look at pathogens, but we can also start looking at toxicants. And this is something that is more available to us because, as I pictured here in the upper right hand corner, we're now getting technology literally the size of like our hand. This is like the size of a deck of cards that we can test things like mercury exposure. And this is really exciting to me because I'm starting to work on the Peruvian Amazon River, where there's a lot of illegal gold mining. So you can see kind of the bottom, the beautiful pristine Amazon, and then the mined version on the right hand side. And through this gold mining process, they use a lot of mercury. So we can use these handheld devices at source in labs down there to start looking at, are the samples we're collecting, let's say in tamarins, containing mercury, and at what levels. And side by side with that, we can use other cool tools such as the one listed below, which is this little thumb drive that basically can run an entire genome on it. So we can really start manipulating these genetic tools and also use them for epigenetic biomarkers to see how well we can look at environmental exposure, and then also what impact that has on their health as we do these bigger and bigger projects at source. Can you go down one more? Okay, I'm done.
Yeah, so I think both Patty and Caroline talked about projects that indicate that we are concerned about, of course, endangered species and conservation, but how that's directly linked to communities and the environment as well, not just the species we're concerned about. So as an organization, we're definitely trying to make our conservation efforts fit into a One Health approach. And One Health just refers to the idea that human health, animal health, and environmental health are inextricably linked. And any successful or the most successful approach to animal conservation will include preserving biodiversity as a whole. Next slide. And by biodiversity, I just mean the variety of life in any particular ecosystem. And I used to think about disruptions in biodiversity as in a fairly simplistic manner. Like if you remove an apex predator from an ecosystem, all the prey species will then become overpopulated, and they might lead to overgrazing of the plants and maybe infectious diseases and stress and malnutrition. Next slide. But in reality, it's a lot more complicated than this, right? Everything is linked to everything else in multiple different ways. And in addition to the animals in the food chain that we can see, we really have to think about the plants, the insects, microorganisms, the soil, the water, the air, everything in that ecosystem to be able to make really strong conclusions about what's the biggest threats to biodiversity. Next slide. I gave a talk about biodiversity and human health earlier this year to the San Diego Natural History Museum. And when I was researching the talk, I came across a lot of, basically a lot of the biodiversity research going on is emphasizing how it's a service. A healthy ecosystem is a service to people. And if you read this quote, 40% of the world's economy, 80% of the needs of the poor derive from biological resources, the richer diversity of life, the greater opportunity for medical discoveries and economic development. I can't get into all of the reasons, all the things they talked about, but one of the most obvious and probably impactful is the amount of biopharmaceuticals that we currently utilize, the number of drugs that are based on studying biological compounds. And if you imagine how many more out there could be ripe for studying and using to our benefit, it's fairly mind-blowing, I think. And I also think that with the way technology is these days, we could discover and study these compounds, but when it came to scaling them up for use, we could do that in a synthetic way, so we don't have to overexploit the natural resource in order to get the benefits of it. The other area that you'll find a lot about in biodiversity is the massive socioeconomic and political implications of biodiversity loss. Next slide, Patty. A good example I watched a talk from a researcher at Berkeley talking about overfishing of the seas, right? This is a huge problem throughout the world. Fisheries are being depleted in many areas and many oceans. And this is obviously not good for the fish populations that are being overexploited, but it's also detrimental to the entire aquatic ecosystem that depends on those fish. Next slide. If you consider the groups that depend on these fish as well, that don't have massive fleets of commercial ships, when these local sustainable fisheries are affected by this, you can go to the next slide. People tend to search out alternative protein sources, right? And this can lead to increases in bushmeat trade, where now additional species are at risk of overexploitation that may be far removed from the original problem, which was in the ocean. And increasing the human-wildlife conflict is a major downstream negative result of biodiversity loss, but it's also obviously contributing significantly to the loss of biodiversity by overexploiting other species. So this is bad for animals, it's bad for the ecosystem. The main point I want to make here was that one major way all of these pressures on biodiversity can affect people is in the form of infectious diseases. Next slide. When habitats are fragmented and species are stressed, infectious disease dynamics are thrown out of balance. And if you plot people right in the middle of that imbalance, or right at the interface of that imbalance, the chances of infectious disease spillover from wildlife to humans becomes a big concern. Next slide. And obviously it can become substantially more than a concern, and nothing's more evident than that right now than the pandemic that we're in. At the same talk, the biodiversity talk, I did talk a little bit about the coronavirus. And at the time, this was at the beginning of February, I had to ask for a show of hands as to who had heard of this virus. And then I had to ask for a show of hands as to who was concerned about it. And not one hand went up in the audience saying that they were concerned. And it's just kind of mind blowing to think that that was just earlier this year, about 10 months ago. So anyway, the last 3 coronavirus outbreaks have come from wildlife. HIV, Ebola virus, swine flu, bird flu, these are all come from wildlife reservoirs. And many of these issues, it's likely that harvest and trade of wildlife species is what creates the original interface between people and these wild animals. So that's not only a major threat to endangered species and thus an imminent conservation concern, but it's also a major public health concern. And there's some regulation on international wildlife trade based on how endangered a species is, but there's very few countries that use strict import controls, and there's really no global regulation on screening for pathogens that are associated with international trade in wildlife. So studying potential emerging diseases before they emerge at these human-wildlife interfaces is generally pretty limited. Next slide. And it doesn't really matter if you're talking about an interface represented by a fairly intense situation, such as a wildlife market where there's lots of different species and high density of people coming into contact with them, or you're referring to a more remote area like Caroline talked about in the Peruvian Amazon, where you have deforestation of pristine jungles for the purposes of development or farming or whatever that may be. As we kind of fragment and chop up wild habitats, we're encroaching on the animals in them. This makes them more crowded, it makes them more stressed, and this facilitates infectious disease transmission. And it can drive the emergence of new infections that not only affect their populations, but again, usually at these interfaces, you're gonna have people. And with people come pets and livestock, and they often bring pests, right? So you're creating a really susceptible pool of people right after disrupting the environment, and that really facilitates spillover into infectious diseases. Next slide. So I didn't want to talk too much about coronavirus just because none of us are currently working on it ourselves, but I did want to put it in the context of conservation relevance. So you can see how such an imminent public health threat like a pandemic that we're in is linked to conservation of endangered species. And that's exactly why we as a group and as an institution as a whole want to have a more holistic approach to conservation through a One Health initiative, making sure that preserving biodiversity as a whole is what's at the core of our initiatives. And I just want to round this out by saying, you know, I said that our mission statement in our group is removing disease as a roadblock to conservation. And you can see the interrelationship between diseases of wildlife, domestic animals, and people. There's lots of complicating factors that interconnect these and come into play when this gets thrown out of balance. And screening for pathogens and doing disease investigations are obviously critical to trying to prevent diseases at the human-wildlife interface. And if this interface involves the trade of endangered species, there's a big niche for people whose focus is on conservation to play critical roles in preventing these problems in the future. So hopefully that's a good enough glimpse into what we can cover in 45 minutes to an hour about what we're up to and what we're about. But I do want to hand it off to a current ARCS scholar to introduce himself and talk a little bit about his research. I think it's a nice segue. So Kyle, if you want to jump in.
Yeah, sure. Hello, everyone. My name is Kyle Malter. I'm a current ARCS scholar. I want to thank you for having me and letting me share my research with you today. Today I'm going to be talking just briefly, just maybe like 5 minutes, about give an introduction to what we study at the Shikuma lab. And sort of in contrast to what we've been talking about, our lab focuses on studying beneficial bacteria and how beneficial bacteria interact with their animal host. And our lab specifically is very interested in the interaction between the bacteria and the host and how that interaction takes place. So a little bit of background about myself. I went to UCLA as an undergrad. I studied biochemistry. And after I graduated, I worked for about 1.5 years in heart research labs. And then after finishing a rotation in Dr. Nicholas Shikuma's lab, I absolutely fell in love with bacteria and sort of the way that they work and their complexity. And so I'm going to be sharing with you a little bit of the work I've been doing in Dr. Shikuma's lab. So if we can go to the next slide. So our lab, we're very interested in understanding how bacteria can beneficially interact with animals. And so the two pictures below are just two sort of common examples that you might have heard about. One is the human gut microbiome. So we now know that there are lots of bacteria that are in our body at any given time that live there happily and that interact with us in a non-pathogenic or non-disease context. And currently, very little is known about how these bacteria could potentially provide any sort of health benefits to us or could be beneficial to us in sort of any context. And that's mostly due to the difficulty in studying beneficial bacteria. So when you're thinking about how do you study beneficial bacteria, it becomes this very difficult question because it's not easy to go in and remove those beneficial bacteria individually without removing other bacteria that live off those bacteria or without removing and allowing for pathogens to take their place. So one of the ways we study that is by using a simplified pair-down model system. And we actually use marine invertebrates. And so on the left here, you can see this is a coral reef. And so corals, when they go through their normal life cycle, they actually require bacteria in order to complete their normal life cycle. And so if we can go to the next slide, I'll talk a little bit about that. So I'll just talk about a few other cases of where we see bacteria interacting with animal hosts. So there's another lab that studies multicellularity in the beginnings of animals, multicellularity, and they found that these choanoflagellates that you see pictured here on the left actually require bacteria in order to form these rosettes and what they believe to be should give us a glimpse into how initial multicellularity occurred, as well as marine invertebrate metamorphosis such as corals, sea urchins, and I'll be talking about tube worms today. That's the model our lab studies, as well as tissue development. This is the squid right there you see. One of its organs actually requires bacteria in order to form, as well as in mammals, we know educating the immune system, bacteria play a major role in that. So if we can go to the next slide. So as I mentioned, the question we're interested in is how do you study beneficial bacterial interactions with their host? So we can go to the next slide. Okay, so how our lab studies that is we use a marine tubeworm model system. So in order to get down to the nitty-gritty of what bacteria are doing to benefit an animal, we need to know how bacteria are interacting with that animal and what they're doing to that animal. So one way to simplify this process is to use one single bacteria and one single animal. So our animal is this marine tubeworm. They are a bottom-dwelling marine organism. When they reproduce, they release their gametes into the water column, or their eggs. Those eggs are fertilized, and then the free-swimming larvae needs to find a place to settle. So if we can go to the next slide. That requires a specific cue. If we go to the next slide. That cue then signals them to metamorphosis and complete their life cycle transition. And what are these cues? We can go to the next slide. So these cues are actually bacteria. So that's why we use this system. So these organisms require bacteria, and that allows us to really probe genetically and using cell biology techniques as well as pharmacology to really understand how these bacteria are interacting with their animal hosts. So one of the key techniques we use to study how bacteria are sort of inducing this metamorphosis, as we like to call it, or providing this life history transition of this model organism, is we use molecular genetics. So molecular genetics, if you want to think of it sort of as, I like to use the analogy of sort of, if you're trying to figure out how a car works, if you sort of take apart the car piece by piece, and looking at what happens to the car when you remove those pieces, that's sort of how we use molecular genetics. We take apart the bacteria, a lot of different genes individually at a time, and then look for a resulting phenotype or a readout. In this case, we look for our worms no longer being able to grow into adults. So if our worms can't grow into adults, we know we've affected the bacteria in such a way that they are no longer providing this benefit of the transition of metamorphosis. So when we did this, we actually found a really interesting, completely novel new structure. So if we can go to the next slide. So we use a single bacteria, Pseudoaltermonas luteoviolacea. And what we found is this bacteria actually produces these viral-like particles to induce metamorphosis of the tubeworm. So what you're seeing now, if we go to the next slide, you can see it in two different images. These are images from our collaborators. These are a viral-like structure that is actually produced by the bacteria that induces metamorphosis. So this structure produced by the bacteria alone is able to induce metamorphosis of our marine tubeworm. And so my PhD has been looking at this structure and trying to understand how the bacteria, or what this structure is doing to the animal in order to induce metamorphosis. And what we're hoping that'll allow us to do in the future is, one, apply this to other marine invertebrates such as corals, since our bacteria is also known to induce metamorphosis of corals, but also apply it to other systems where we find this structure found as well. So this structure has also been found in the gut microbiota of humans, but there's currently no studies looking at what it does or how it functions. So what we're hoping to do is apply the studies that I'm currently working on to those studies as well. So I'm just going to give a quick, very, very quick introduction to the science that I'm doing. So if we go to the next slide. So this is just a little model that I put together where we've published 2 papers on this structure looking at how the bacteria produce this structure. We've actually identified 2 effector genes, effectors meaning genes that are released from this structure into the animal host, our marine tubeworm, that then perform some function. A lot of my expertise in biochemistry and cell biology has allowed me to really suss out sort of the nitty-gritty of how these proteins are functioning and what they're doing in the animal host. So if we go just real quick to the next slide, I just wanted to, so you can see this is a model system that our lab has been able to recreate. And this is sort of what we've been trying to understand, is just sort of the very nitty-gritty molecular cell biological process of these proteins, as well as this structure in inducing metamorphosis. So if we can actually go back a few slides, back to the marine tubeworm slide. Yeah, maybe one more. Yeah, right here. So what we're hoping to do is then use this knowledge of these structures, and hopefully we can apply it and add it to the tools. Another researcher in our lab is actually working with corals, hoping to work with corals this next coming year. This last year, I think the COVID pandemic kind of slowed us down, but we're hoping to work with corals and study coral reef restoration and how these structures might be used to benefit coral reef restoration laboratories, and add it to their toolbox of mechanisms for doing coral reef restoration in situ or at the site. And then, yeah, so that's a very brief overview of what our lab does. And with that, I just want to, if we can go to just to the last slide, I just want to thank my lab members. I just want to thank my lab members and Dr. Nicola Shikuma, the Milagros Esmeralda, she's kind of tacked on there on the right. She was working very closely with my undergrad and contributed majorly to my work. So I just want to thank them, as well as the ARCS scholars for funding me. It means a lot to us and my family. I actually have my daughter here. I just thought I'd show her off to you guys. She's hi. Yeah, so the funding at ARCS really means a lot to me and my family, and I want to thank you guys so much for supporting me in my research. And with that, I guess we'll open up to questions, I believe.
Please, if you have questions, submit them in the chat box so that we don't have too many people talking at once. And I will start, first of all, by thanking our presenters. It was truly an exceptional presentation. And we're so proud of you. You make us, everything that we do at ARCS is deeply rewarded by what we hear that you're doing. We did have a couple of early on questions, one very germane. How would you as the scientist talk to the lay public about the importance of biodiversity and conservation? We need to make that important work, the importance of conservation known in non-science talk, and just regular speak.
It's really hard to answer. It's something that we deal with every day, is trying to translate what we're doing, because working at the zoo, a lot of the messaging that goes out is going to the general public. We of course have ways of communicating with our scientific colleagues, but what we really want to do is communicate and educate the public and non-scientists. As far as biodiversity goes, to me, it just using real life examples is the easiest thing to explain. I think one area is biomimicry, where people study natural things and then create synthetic versions of it that make our lives better. So I was reading about how certain species of sharks, the way their skin, just the structure of their skin prevents bacteria from adhering to their skin. And there are scientists who have made this film that mimics the structure of shark skin. I think they just 3D printed it, and they're trying to use it as surfaces in hospitals. Because as you know, nosocomial infections or hospital-acquired infections are a huge, huge, huge problem. And if they can just prevent the amount of bacteria growing on surfaces, that could be huge for diseases in hospitals. And there's a million. There was a whole field of biomimicry. You can look it up. Actually, if you want to go into a rabbit hole of cool videos and stuff, just look up biomimicry, because it's amazing. And then of course, just saying, like, antibiotics, most contemporary antibiotics derived from studying a fungus and a product produced by a fungus that has antibacterial effects. And now we have all these drugs based on that, penicillins. And then one of the more common chemotherapeutic agents that's used right now for, I think, breast cancer and ovarian cancer comes from the bark of a tree, the Pacific yew tree in California. These are really prominent examples, but again, if you think of all the frontiers we've yet to characterize, all the chemical compounds in nature and what we may be able to get from those in treating human diseases, preserving that is huge, because you can really mess with biodiversity very easily. Humans can, through overdevelopment and overexploiting the natural resource. It's still a little technical, but yeah, I think just being able to use real life examples that people deal with every day or are close to home for them is one way of trying to translate the message.
And I think we sometimes we do struggle as veterinary pathologists because what we deal with is death, and what we try to communicate is our findings associated with postmortem exams. But the way that I like to present that and discuss it is that we basically have the opportunity and actually the privilege of providing the last entry into the medical record for every animal that that comes through the zoo, and we take that responsibility seriously. And as alluded to during the presentation, the samples that we collect and the information that we provide goes into an archive. And I think everyone can have sort of appreciate and get an understanding when you say we have 55,000 cases in our archive, and that we can go back and utilize that information and provide specimens to museums. And so sometimes framing what we do as part of the medical documents that go along with these animals and will exist forever is one way I think we get past that point of just discussing these as cases of animals that died, but that are contributing to research in perpetuity.
Yeah, I think it's a great question. I was recently asked this question because we were working with these San Bernardino kangaroo rats, and they're, one subspecies of all these kangaroo rats. So why do we care that there's only 30 left? And at first it's hard to answer. Like, would anyone notice if one little rodent disappeared? But I think just building off what Steve and Patty said, and that each animal does have a role, even if we don't know what it is yet, and we don't know how that's going to impact our life moving forward and making it better. That's just one reason. That's all of the reason I think we need to protect them rather than trying to bring them back from extinction.
Sometimes I just say, look, we're talking about air and water. You don't need to get into hardcore science to say we drink water and we breathe air, and when you disrupt these ecosystems, all of that is affected. And it may not be immediately obvious, but if you look at trends where water quality and availability of things like water are going, and air quality in areas where there aren't really any regulations, it's a significant problem. And it's hard to get buy-in because a lot of it tends to be preventative, and it's hard to invest in preventative measures because the problems maybe haven't happened yet. It's hard to get people on board with things that aren't an immediate crisis. So yeah, it's a complicated problem. It's a really complicated problem. And on top of that, there's a lot of people with pretty solid credentials saying some really wild stuff on the internet and in other forums that make it really frustrating. People with degrees and prominent alma maters that are really muddling up messages about things like this, from everything from climate change and habitats to vaccines and infectious diseases, and it's a constant challenge.
Good answers. Thank you very much. You were wonderful in showing how the San Diego Zoo global research goes out to other countries in the field. How was that funded?
Our field programs, especially those internationally, there's a wide variety of ways that they're funded. So, for example, Stephen Chege, who's our postdoctoral fellow in Kenya, he's funded by an internal postdoc called the Company 9 postdoc, which is funded by the Institute for Conservation Research specifically. Larger initiatives like building a laboratory often benefit from donor support. So we have gotten donor support for the laboratory that we're building at Lewa, both for the construction of the laboratory and for equipment that we want to bring into the laboratory. And then we also partner with the conservation organizations in the country to help with fundraising. So one of our big partners in Kenya is the Northern Rangelands Trust, and they oversee over almost 40 wildlife conservancies in Kenya, and we work in tight, sort of in lockstep with them with fundraising efforts. And the efforts are not just for animal care and disease diagnostics, but also to employ rangers to help with anti-poaching efforts, to build fences, to protect sanctuaries. We also have a veterinary technician based in the Horola Conservancy, and we fund his salary. So I would say it's a variety, and then also grants. Depending on the study, grants are written to both international agencies or US-based agencies to fund a lot of this work. So it's really a multi-pronged approach to providing funding for the various aspects of the work that we do.
Follow-on question to that. Has that changed at all since the emergence of Ebola? Pardon me, or SARS. I meant SARS-CoV, but I said Ebola. Either one of those.
Well, I think that the interest internationally is really targeted at disease surveillance. So if anything, I think it has increased our ability to fundraise in these areas, and it has increased awareness for the importance of disease surveillance efforts, not just laboratory-based, but as both Caroline and I talked about, these in situ decentralized laboratories that are based at the source of infections and really at the human wildlife livestock interface where these spillover events are more likely to occur. So I think, if there's, I won't say that, anything good has come from COVID, but it has raised awareness for the kind of disease surveillance work that we do, and that allows us to do more of it.
I want to remind the audience that in the invitation, there was a hyperlink to our 3 alumni from the San Diego Zoo. So if you have more specific questions or interests in funding, you can contact them directly from the invitation. Another question we had is that have we ever used phage therapy, bacterial phage therapy, to combat bacterial infections in the wildlife? I mean viral phage therapy to combat bacterial infections in the wildlife. This ARCS chapter recently was introduced to the power of phage therapy in humans by a new member, Stephanie Strafty, who had a book out and some very interesting work, interesting presentation that I think is going into a movie.
Not that I know of. I think it's managing diseases, especially in the wild populations, is heavily on prevention. It may be an area, especially for the clinical veterinarians working in the zoo and safari park, that are constantly, they have a lot more emphasis on individual patient care, where things like antibiotics are always in discussion, using products that are effective without having to overprescribe antibiotics, right? They don't want to have issues with antibiotic resistance. They're always open to discussing new therapies, and they're always they have their fingers on the pulse of these kinds of things. But I would say the area of zoo and wildlife medicine tends to be the end of the line with things, and in some ways that's unfortunate because of how much we care about it. But at the same time, there's some ethical issues to using novel therapies for particularly an endangered species where it's not been proven to be safe. And there's a little bit of hesitance, I think, in some people's minds to extrapolate safety studies that were maybe performed on mice or experimental rats or something like that, and extrapolating that to use for some of the endangered species that they treat on a day-to-day basis. But the veterinarians that we work with they're state-of-the-art people. They're always up for learning about new potential therapies. So off the top of my head, since I don't do that, I don't know if they've even heard about it or what kind of avenues they have for pursuing utilizing novel therapies. But I will ask, because that's an interesting question.
You gave a slide that showed some of the little handheld molecular diagnostics that can be used in the field. And so many zoo researchers go into the field, into these remote areas, and capture and collar and then release animals so that we can track their behavior. At the same time, when you collar them, do you also use handheld molecular diagnostics to see whether they're carrying diseases that could be potential epidemics?
Yeah, that's, I can speak to that. That's one of our main goals with this project that we're collaborating with several field groups, including Field Projects International, which has been a group that has been collecting samples for over 10 years on the Peruvian Amazon. And so they already have a system in place, and we're working not only for disease investigations, but also testing out some of these new tools, and then also looking into novel conservation technology to help track these critters. So, you know, what the device, you know, that we use to capture the animal in the first place so that we can handle it and take the samples and release, can we devise something that maybe the animal gets a little, you know, a little tracker, a little backpack, and that maybe this cage that's out there, you know, they don't get locked into it. They just walk past, they happen to go on a scale, their little scanner gets scanned, they get a picture taken, maybe they rub against a thing that collects some fur or hair, you know. So we're building ways that we can just really minimalistically test them. And then, like you said, at the time, we take those hair samples and run them through this mercury tester or something like that at source. And there's a little bit difference between, you know, what can we do right in the field versus what can we get back to a lab, you know, a couple miles away, and then what can we do in our lab facilities here. So we're looking all along that pipeline.
Another example of a handheld device that we plan to deploy, but again, COVID has slowed us down a little bit, is a handheld PCR machine that we are intending to use with the orphan elephants at Ritteti. Based on our EEHV work here at the zoo with our own herd, what we actually want to be able to develop is a test that would be totally non-invasive for the elephants at Ritteti by using the saliva that is on the tips of the bottles that they feed from. And so we don't want to train those animals for any kind of sample collection, because the intent is for them to be released back to the wild. So we don't want them habituated to people, and we don't want to train them for intense sample collection like we have in our own herd here, but we have two sources that are abundant, and that is saliva and poop. And so if we can validate our field assay for EEHV, we can get those samples from those elephants and actually track in real time their shedding patterns, because we suspect that, you know, they come in, they're under stress, and this is the time when a disease like a virus like herpes virus can recrudesce. And so if we can deploy that device in Kenya, we can learn a lot about the natural history and shedding patterns of herpes virus there.
Great answers. I want to be aware of everybody's time here. We're actually over time right now. If there's any last question, let us know. In the meantime, let me turn it back over to Holly, and Holly can see if a last minute question pops up, but I want to make sure that we turn it back over to her for final comments.
Thank you, Anne. I can't tell the three of you, plus Kyle, the four of you, how we are that you were able to come and speak to us today. It is so impressive to see the work you're doing and to hear about your careers and your plans for the future. It makes me proud to be part of ARCS when I see the kinds of work that we support and that you so much appreciate. And I think, Steve, it was you that pointed out the wonderful nature of our ARCS awards, which are unrestricted, and I know that you scholars really do appreciate that. And you know, you guys are doing so much. You are tomorrow's leaders, and we're just so pleased that you could spend time with us today to tell us about your schooling, your discoveries, your research. And I would love to come back and have you speak to us again, hopefully in the real world when we're finished with COVID. I'd also like to thank Anne for a wonderful job of introducing and moderating everyone. And also Margaret Dudas and Janet Romer, who are part of this committee and put today's event together. I'd also like to especially thank Sherry Lundeen, our national president, for attending, as well as Joe, Barb, and Leslie. coming as presidents from 3 of our other chapters in the country. I think it's great. You know, that is one of the silver linings of a terrible tragedy, the COVID pandemic, is that we are in this position to not only have a field trip virtually and learn so much from you 3 and 4 scholars, but also to share this with not just the San Diego group and chapter, but with people all over the country. And Rob, thank you for recording it and being a part of it, because I know people already have expressed an interest in seeing the recordings. So I'm just so thankful and appreciative to all of you for what you do. And thank you for being here today. I don't see any more questions unless anybody has some that they would like to ask and get off mute and ask right now.
A quick question, Holly. How will we find the link to the recorded presentation for those people who have dentist appointments and couldn't be here with us today?
Rob will answer that, please.
Yeah, so we can make that available, the information on the recorded version online. So just go to the website and it'll be there under events.
Under the San Diego ARCS website.
Correct.
Yeah, okay. And I'm sure Ingrid will be putting out some social media information about it as well. And I wanted to point out, as a special coincidence, Ingrid Benershka Perkins is one of our members, and we all know that San Diego Zoo Global and the whole conservation and reproduction aspects of the zoo are due to her father, Dr. Kurt Benershka. And so it's really a pleasure and an honor to have Ingrid as one of our members as well. Thanks, Ingrid. And thanks, everybody, and have a great Monday.
Thank you.
Thank you all.
Thank you.
Bye-bye.